Imagine for a moment the single most requested goal in the world of dermatology. It isn't just about curing a specific disease, it is about reversing time itself. We are talking about the quest to heal aged skin, specifically skin that has been damaged by decades of exposure to the sun. For years, we have had a gold standard ingredient. You've probably heard of it. You might even have a bottle of it sitting in your bathroom cabinet right now. It is called retinol. It is the closest thing we have to a time machine for our cells. But there is a catch, a very big, very red, and very painful catch. For millions of people, this miracle ingredient is unusable. Instead of glowing, youthful skin, they get what is often called the retinoid uglies, severe redness, peeling, burning, and irritation. It is a reaction so common that it causes huge numbers of people to give up on the treatment entirely. But here's the part most people don't realize. We still don't fully understand exactly why this irritation happens at the molecular level. We know that it happens, but the precise biological pathway, the traffic signals inside your cells that turn a helpful treatment into a painful rash, is complex. Today, we are going to dive deep into a specific clinical research study that aims to solve this mystery. Researchers set out to look not just at the surface of the skin, but deep inside the cellular machinery. They wanted to understand the relationship between how much retinol you use, how often you use it, and the genetic signals that trigger inflammation. This is not just a story about a beauty cream. This is a story about the delicate balance between healing our bodies and hurting them. This is the science of retinol-induced dermatitis. To understand why this specific research trial is so important, we first have to understand the sheer scale of the problem it addresses. Skin aging is not just a cosmetic concern, it is a biological decline. There are two types of aging. First, there is intrinsic aging, the natural, slow clock ticking inside us that makes our skin thinner and more fragile as we get older. But then there is extrinsic aging, often called photoaging. This is damage caused by external factors, primarily ultraviolet radiation from the sun. When UV rays hit your skin, they don't just cause a tan, they trigger a cascade of destruction. They activate enzymes called matrix metalloproteinases, which you can think of as tiny molecular scissors.
These scissors go around cutting up the collagen and elastin fibers that give your skin its structure and bounce. Over years, this results in deep wrinkles, leathery texture, and pigmentation changes. Currently, topical retinoids, which are derivatives of vitamin A, are the only medical therapy proven to repair this damage. They are not just moisturizers. They are biologically active drugs that reprogram your skin cells. They tell your cells to speed up turnover, to produce more collagen, and to stop breaking down the existing structure. However, this power comes at a cost. The side effect profile of retinoids is significant. The condition is clinically known as retinoid dermatitis. It involves erythema, which is medical speak for redness, along with scaling, dryness, and a burning sensation. Why does this matter? Because in medicine, a treatment is only as good as the patient's ability to stick with it. If the only cure for a condition makes you feel miserable or look worse in the short term, you stop using it. This is known as non-compliance. In the context of treating aged skin, this barrier is huge. If researchers can figure out exactly how to separate the anti-aging benefits from the inflammatory side effects, it would revolutionize dermatology. It would mean we could treat skin damage effectively without the burn. And this is where the study we are discussing today comes in. It wasn't designed to just test a new product. It was designed to investigate the biology of the reaction itself. Before we look at the study design, we need to understand the mechanism these researchers are exploring. When you apply retinol to your skin, it doesn't work immediately. Retinol is actually a precursor. Your skin cells have to convert it first into retinaldehyde, and then into the active form, which is called retinoic acid. Retinoic acid is the key that unlocks the door. It enters the nucleus of the cell and binds to specific receptors on your DNA, known as retinoic acid receptors. This binding turns certain genes on and others off. It is literally changing the instructions your cells are following. The researchers in this study were interested in a specific paradox. We know that retinoic acid activates genes that repair collagen, but it seems to also activate pathways that cause inflammation. One of the specific pathways mentioned in this research involves the EGF receptor, or epidermal growth factor.
This is a protein on the surface of cells that helps control cell growth and division. When this pathway is overstimulated or dysregulated, it can lead to chaotic skin behavior. The hypothesis here is that the irritation we see, the redness and peeling, isn't just a random chemical burn. It is a specific, programmed biological response involving the metabolism of vitamin A and the activation of these growth factor pathways. If the researchers can map this pathway, they can answer critical questions. Does the irritation depend on the dose? Does it depend on how frequently you apply it? And most importantly, can we see the irritation coming in the genes before we see it on the face? So, what exactly is being tested in this clinical trial? The core objective is to investigate the dose response and time dependence of retinol-induced dermatitis. Let's break that down. Dose response asks a simple question. If we double the amount of retinol, do we double the irritation? Or is there a threshold where the skin suddenly reacts? Time dependence asks, when does the reaction peak? Is it after 2 days, 2 weeks? Does the skin eventually get used to it? A process we call retinization. But here is where things get interesting. The researchers aren't just looking at the skin with their eyes, they are looking at it biochemically. They are testing the hypothesis that we can measure the level of irritation by looking at biochemical markers. These are specific molecules or genetic signals that appear in the skin when it is under stress. Specifically, they are looking at how retinol regulates retinoid-responsive genes. Remember those receptors on the DNA we talked about? When retinol activates them, they produce specific proteins. The researchers want to measure these proteins to see if they correlate with the visible redness and peeling. They are also investigating the role of that EGF receptor pathway we mentioned. By understanding if this specific pathway is the culprit for the dermatitis, it could open doors for future treatments that block this pathway while still allowing the anti-aging benefits to continue. It is a study designed to deconstruct the molecular machinery of a side effect. Now, let's walk through how a study like this is actually conducted. This is a detailed look at the clinical trial process. This study focuses on an adult population, specifically those with aged skin.
The inclusion criteria would typically look for individuals who have visible signs of photoaging, wrinkles, texture changes, or discoloration, because these are the people who would normally use retinol. The study design is likely an interventional model. This means the researchers are actively doing something to the participants, in this case, applying a topical retinol formulation, rather than just observing them. The participants would be divided into groups or undergo a protocol where they apply specific doses of retinol. The researchers mentioned investigating dose and frequency, so it is highly probable that different participants applied the cream at different strengths or on different schedules, perhaps once a day versus every other day. But how do they measure the results? This is the most critical part of the trial design. First, there are the clinical assessments. This is the standard dermatological exam. A trained clinician examines the participant's skin and scores it on a scale. They are looking for erythema, the intensity of the redness; scaling, is the skin flaking off; edema, is there any swelling; subjective symptoms, they would ask the participant to rate their pain, itching, or burning sensation. However, clinical assessment is subjective. One doctor might rate redness as a 5, while another rates it as a 4. To get hard data, this study goes deeper. This leads us to the biochemical assessments. This usually involves skin biopsies. A biopsy is a procedure where a small sample of skin is removed, usually with a punch tool, under local anesthesia. It sounds intense, but it is the only way to see what is happening inside the tissue. Once they have these skin samples, the researchers take them to the lab. They extract the RNA, the messenger molecules that carry instructions from DNA, and perform an analysis to see which genes are being expressed. They are looking for markers for retinoid bioactivity. In simple terms, they are checking to see if the retinol is actually doing its job inside the cell. They are also checking for markers of inflammation. By comparing the clinical score, how red the face looks, with the biochemical score, how active the inflammation genes are, they can build a complete picture of the condition. The study also aims to look at the time dependence. This means these assessments aren't just done once. They would be done at baseline, before treatment starts, and then at specific intervals, perhaps 24 hours later, 4 days later, or 2 weeks later. This allows the researchers to plot a timeline of irritation. But here's the part that really makes you wonder. We often assume that more is better. If a little bit of retinol helps your wrinkles, surely a lot of retinol will help them faster, right? This study challenges that assumption. There is a theory in toxicology called hormesis, where a substance can be beneficial at low doses, but toxic at high doses. In the case of retinol, there might be a sweet spot, a specific concentration where the genes for collagen repair are fully activated, but the genes for inflammation and dermatitis are not yet triggered. If this study can identify that sweet spot, or identify the specific day when the skin's barrier breaks down, it changes everything about how we prescribe these products. It suggests that the burn isn't necessary for the beauty. It suggests that the irritation might just be an overdose reaction that we have normalized for decades. So, what are the implications of this research? Let's look at the potential outcomes. The best case scenario: if the researchers successfully identify the specific genes in the EGF receptor pathway that cause dermatitis, and if they find that these genes are separate from the ones that cause anti-aging, the pharmaceutical industry could develop smart retinoids. These would be molecules designed to bypass the inflammatory pathway entirely. Imagine getting the results of prescription-strength tretinoin with the gentleness of a basic moisturizer. The neutral scenario: the study might find that the two pathways are inextricably linked. It might turn out that the inflammation is actually part of the healing process, that you need a little bit of injury to stimulate the repair. In this case, the research would still be valuable because it would help doctors optimize dosing schedules to keep the irritation manageable rather than eliminating it. The negative scenario: it is possible that individual variation is just too high. The study might find that one person's genes react completely differently than another's, making a one-size-fits-all dosing recommendation impossible. While frustrating, this is also a scientific finding. It tells us that personalized medicine, perhaps based on a genetic swab, is the only way forward.
Regardless of the outcome, this data helps build the pharmacokinetics of skincare. Just as we know exactly how much ibuprofen to take for a headache, we need to know exactly how much retinol to use for aging skin without causing a chemical burn. While researchers are hard at work figuring out the molecular details, what does this mean for the general public right now? If you are struggling with skin aging or considering using these ingredients, what are the current best practices? It is important to remember that while retinoids are the gold standard, they are not the only way to care for aged skin. 1. Sun protection. The most effective way to treat photoaging is to prevent it. Broad-spectrum sunscreen is the single most effective anti-aging product available. It stops the UV rays from activating those molecular scissors we talked about earlier. 2. The low and slow approach. Based on the principles of this study, that dose and frequency matter, dermatologists often recommend starting with the lowest possible concentration of retinol. You might apply it only twice a week at first, slowly increasing the frequency as your skin builds tolerance. This gives your cells time to adjust their genetic expression without triggering a massive inflammatory response. 3. Barrier support. Since we know retinol compromises the skin barrier, using rich moisturizers containing ceramides, fatty acids, and cholesterol can help mechanically repair the shield that the retinol is temporarily weakening. 4. Alternative ingredients. For those who simply cannot tolerate retinoids, ingredients like bakuchiol are being studied. Early research suggests bakuchiol may stabilize some of the same collagen-producing pathways as retinol, but with less activation of the inflammatory pathways. 5. Lifestyle factors. Never underestimate the power of systemic health. A diet rich in antioxidants, adequate hydration, and avoiding smoking are foundational. Smoking, in particular, accelerates the breakdown of collagen in a way that mimics photoaging. We must also touch upon the safety and ethics of running a trial like this. Clinical trials are heavily regulated. Before a single participant is enrolled, the study protocol must be reviewed by an institutional review board or an ethics committee. Their job is to ensure that the risk to participants is minimized and justified by the potential scientific gain.
In a study involving skin biopsies, this is crucial. A biopsy is a minor surgical procedure. It carries risks of scarring, infection, and pain. Participants must provide informed consent, meaning they are told exactly what will happen to them, what the risks are, and that they can withdraw at any time.
Furthermore, applying an ingredient known to cause dermatitis means the researchers are expecting to cause harm, mild harm in the form of a rash, but harm nonetheless. Ethical oversight ensures that if a participant has a severe reaction, there are medical protocols in place to treat them immediately. This slow, careful process is why medical research takes time. We cannot just test chemicals on people without strict guardrails. Every data point in this study represents a volunteer who agreed to undergo discomfort to help advance our understanding of human biology. To summarize, the clinical trial we explored today is a fascinating journey into the molecular depths of our skin. We learned that researchers are not satisfied with just knowing that retinol works, they want to know how it works and why it hurts. We discussed the battle between retinoic acid receptors and the EGF receptor pathway. We looked at how scientists use skin biopsies to read the genetic code of inflammation. And we explored the hope that one day we might decouple the beauty benefits from the burning side effects. This study reminds us that the skin is not just a wrapper for our bodies. It is a complex, living organ with its own immune system, its own communication networks, and its own way of telling us when it is overwhelmed. As science progresses, we move closer to a future where skincare is not just about trial and error, but about precise, genetically informed treatments that work in harmony with our biology. If you found this deep dive into the science of clinical trials interesting, there is so much more to uncover. The world of medical research is vast, with thousands of studies happening right now that could shape the future of health and longevity. We will continue to track these developments, breaking down complex protocols into clear, understandable insights. If you want to stay informed about how the medical world is evolving, consider subscribing to the channel. It helps us keep creating deep, educational content like this. Thank you for watching and for being curious about the science behind the surface.
Disclaimer: this video is for informational purposes only and does not provide medical advice. The content is based on publicly available data from clinical trial registries and is intended for educational use.